Eukaryotes. Eukaryotes are organisms made up of cells that possess a membrane-bound nucleus or that holds DNA in the form of chromosomes, as well as membrane-bound organelles. Eukaryotic organisms may be multicellular or single-celled organisms. All animals are eukaryotes. Other eukaryotes includes plants, fungi, and protists. A typical eukaryotic cell is surrounded by a plasma membrane and contains many different structures and organelles with a variety of functions. Examples include the chromosomes, or a structure of nucleic acids and protein which carry genetic information in the form of genes, and the mitochondria, often described as the powerhouse of the cell. Organelles that are commonly found in eukaryotic cells. Nucleus, the brains of the cell. The nucleus directs cell activities and contains genetic material called chromosomes made of DNA. Mitochondria make energy out of food. Ribosomes make protein. Golgi apparatus make process and package proteins. Lysosome contains digestive enzymes to help break down food. Endoplasmic reticulum, called the intracellular highway, because it is for transporting all sorts of items around the cell. Vacuole, used for storage. Vacuoles usually contain water or food. Prokaryotes. Prokaryotes are organisms made up of cells that lack a cell nucleus or any membrane-encased organelles. This means the genetic material DNA in prokaryotes is not bound within a nucleus. In addition, the DNA is less structured in prokaryotes than in eukaryotes. In prokaryotes, the DNA is a single loop while eukaryotes' DNA is organized into chromosomes. Most prokaryotes are made up of just a single cell or unicellular, but there are a few that are made of collections of cells or multicellular. Scientists have divided the prokaryotes into two groups, the bacteria and the archaea. Some bacteria including E. coli, Salmonella, and Listeria are found in foods and can cause disease. Others are actually helpful to human digestion and other functions. Archaea were discovered to be a unique life form which is capable of living indefinitely in extreme environments such as hydrothermal vents or Arctic ice. A typical prokaryotic cell might contain the following parts. Cell wall, the membrane surrounding and protecting the cell. Cytoplasm, all of the material inside the cell except the nucleus. Flagella and pili, protein-based filaments found on the outside of some prokaryotic cells. Nucleode, a nucleus-like region of the cell where genetic material is kept. Plasmid, a small molecule of DNA that can reproduce independently. Reproduction of organisms and their cells. Prokaryotic cell reproduction. Prokaryotic cells reproduce by a process known as binary fission. One cell splits in half to become two daughter cells. Before a prokaryotic cell divides in half, the chromosome must be duplicated. In a broad sense, the science of classification, but more strictly, the classification of living and extinct organisms. In other words, biological classification. The term is derived from the Greek taxis, means arrangement, and nomos means law. Taxonomy is, therefore, the methodology and principles of systematic botany and zoology, and sets up arrangements of the kinds of plants and animals in hierarchies of superior and subordinate groups. Among biologists, the Linnean system of binomial nomenclature, created by Swedish naturalist Carolus Linnaeus in the 1750s, is internationally accepted. Carl Linnaeus, also known after his ennoblement as Carl von Linné, was a Swedish botanist, zoologist, and physician who formalized binomial nomenclature, the modern system of naming organisms. He is known as the father of modern taxonomy. The three domain systems are bacteria, archaea, and eukaryota. The bacteria, also known as eubacteria or true bacteria, are prokaryotic cells that are common in human daily life, encounter many more times than the archaebacteria. Eubacteria can be found almost everywhere and kill thousands upon thousands of people each year, but also serve as antibiotics producers and food digesters in our stomachs. The bacteria possess the following characteristics. Bacteria are prokaryotic cells. Like the eukarya, they have membranes composed of unbranched fatty acid chains attached to glycerol by ester linkage. The cell walls of bacteria, unlike the archaea and the eukarya, contain peptidoglycan. Bacteria are sensitive to traditional antibacterial antibiotics, but are resistant to most antibiotics that affect eukarya. Bacteria contains rRNA that is unique to the bacteria, as indicated by the presence molecular regions distinctly different from the rRNA of archaea and eukarya. Bacteria include Mycoplasmas, cyanobacteria, gram-positive bacteria, and gram-negative bacteria. Archaea or archaebacteria. The archaea possess the following characteristics. Archaea are prokaryotic cells. Unlike the bacteria and the eukarya, the archaea have membranes composed of branched hydrocarbon chains, or many also containing rings within the hydrocarbon chains, attached to glycerol by ether linkage. The cell walls of archaea contain peptidoglycan. Archaea are not sensitive to some antibiotics that affect the bacteria, but are sensitive to some antibiotics that affect the eukarya. Archaea contain rRNA that is unique to the archaea, as indicated by the presence molecular regions distinctly different from the rRNA of bacteria and eukarya. The eukarya or eukaryotics. The eukarya possess the following characteristics. Eukarya have eukaryotic cells. Like the bacteria, they have membranes composed of unbranched fatty acid chains attached to glycerol by ester linkage. Not all eukarya possess cells with a cell wall, but for those eukarya having a cell wall, that wall contains no peptidoglycan. Eukarya are resistant to traditional antibacterial antibiotics, but are sensitive to most antibiotics that affect eukaryotic cells. Eukarya contain rRNA that is unique to the eukarya, as indicated by the presence molecular regions distinctly different from the rRNA of archaea and bacteria. Determining relatedness among organisms. Scientists can now determine relatedness by comparing the frequency of nucleic acid bases, or the G plus C content, perform nucleic acid hybridization to determine the degree to which DNA sequences are the same, perform nucleic acid sequencing, or perform other advanced molecular techniques. But in the past, the only way we had to determine relatedness between organisms was morphology, or the observable structure of the organisms. Very often, molecular techniques will give us the same results as morphological techniques. A cellular infectious agent, bacteria domain, and archaea domain. Microbes within the domain bacteria and archaea are all prokaryotes, or their cells lack a nucleus, whereas microbes in the domain eukarya are eukaryotes, or their cells have a nucleus. Some microorganisms, such as virus, do not fall within any of the three domains of life. According to this system, the tree of life consists of three domains: archaea, bacteria, and eukarya. The first two are all prokaryotic microorganisms, or single-celled organisms, whose cells have no nucleus. Diversity of microorganism. Diversity of microorganism. The ability of soil microorganisms to decompose organic matter is a function of their population complexity. The diversity of microorganisms enhances the capability of soil to degrade a wide variety of organic substances. Some prey on pathogenic bacteria and reduce the survival of viruses. Diversity of microorganisms. Bacteria, or Cynebacterium, include two groups: Eubacteria, peptidoglycan cell walls. Archaebacteria, lack of peptidoglycan cell walls. Shapes, several forms: bacilli, rod-like or Cynebacillus. Cocci, spherical or Cyncoccus. Spiral, corkscrew or curved. Square, star shape. Microbial physiology. Microbial physiology is defined as the study of how microbial cell structures, growth, and metabolism function in living organisms. It covers the study of viruses, bacteria, fungi, and parasites. Metabolism. Metabolism, the sum of the chemical reactions that take place within each cell of living organism and that provide energy for vital processes and for synthesizing new organic material. What are the two types of metabolism? Two types of metabolic reactions take place in the cell: building up, or anabolism, and breaking down, or what you call catabolism. Catabolic reactions give out energy. They are exergonic. In a catabolic reaction, large molecules are broken down into smaller ones. An example of metabolic reaction is the one that takes place when a person eats a spoonful of sugar. Metabolic process. First, food intake, food breakdown, energy created, body repair, and the last is activity energy. Three stages of metabolism. Stage 1, nutrients are digested into absorbable units, into the blood and moved to tissue cells. Stage 2, anabolism, nutrients are made into macromolecules. Stage 2, catabolism, nutrients broken down into pyruvic acid and acetyl CoA, glycolysis major catabolic pathway. Stage 3, CO2 released. H and O molecules make water. Some energy used to make ATP. Citric acid cycle and oxidative phosphorylation major pathways. Bacterial genetics. Bacterial genetics is the study of how genetic information is transferred, either from a particular bacterium to its offspring or between interbreeding lines of bacteria, and how that genetic information is expressed. Given the short generation times of most bacteria, the inheritance of genetic information must be extremely faithful. Occasionally, genetic variation or transfer of genetic information between bacteria gives rise to mutations. The large sizes of bacterial populations ensure that even extremely rare genetic events are likely to occur. This genetic variation makes it possible for individual members of huge populations of bacteria to evolve new traits rapidly. For example, a single mutation may allow a bacterium to survive environmental conditions that would kill its non-mutant siblings. For example, exposure to an antibiotic, or a group of genes transferred from another bacterial species may enable such an altered bacterial species to invade a new environmental niche. For example, the ability to infect a new host. In the laboratory, genetic variation is exploited to study the properties of bacteria, to explore the fundamental characteristics of gene transfer and gene expression, and to construct mutants with desired characteristics. Techniques that have enabled this discipline are culture in defined media, replica plating, mutagenesis, transformation, conjugation, and transduction. Bacterial genetics, asexually reproducing or binary fission, high reproductive rates or short generation spans. Variation in bacteria mainly caused by mutation. What is genetic engineering? Genetic engineering, sometimes called genetic modification, is the process of altering the DNA in an organism's genome. This may mean changing one base pair, AT or CG, deleting a whole region of DNA, or introducing an additional copy of a gene. It may also mean extracting DNA from another organism's genome and combining it with the DNA of that individual. Genetic engineering is used by scientists to enhance or modify the characteristics of an individual organism. Genetic engineering can be applied to any organism, from a virus to a ship. For example, genetic engineering can be used to produce plants that have a higher nutritional value or can tolerate exposure to herbicides. What is gene therapy? Gene therapy is when DNA is introduced into a patient to treat a genetic disease. The new DNA usually contains a functioning gene to correct the effects of a disease-causing mutation. Gene therapy uses sections of DNA, usually genes, to treat or prevent disease. The DNA is carefully selected to correct the effect of a mutated gene that is causing disease. The technique was first developed in 1972 but has so far had limited success in treating human diseases. Gene therapy may be a promising treatment option for some genetic diseases, including muscular dystrophy and cystic fibrosis. There are two types of gene therapy depending on which types of cells are treated. First is the somatic gene therapy. Transfer of a section of DNA to any cell of the body that doesn't produce sperm or eggs. Effects of gene therapy will not be passed on to the patient's children. The second one is the germline gene therapy. Transfer of section of DNA to cells that produce eggs or sperms. Effects of gene therapy will be passed on to the patient's children and subsequent generations. In the future, this technique may allow doctors to treat a disorder by inserting a gene into a patient's cell instead of using drugs or surgery.